Hi, I'm Dr. Rosario, and this video is the history of microbiology. In this video, I'm going to be talking about the major discoveries that led us to where we are today in the field of microbiology. Now, of course, I'm not going to be able to cover everything, but I am going to hit up the high points. I'm going to cover some of those key historical milestones that have framed and really led to where we are now. For your exam, I really want you to focus in on some of the names and the major discoveries associated with those names. I am not going to test you on dates. That is not something that is necessarily important to me. But I do want you to see kind of how we progressed from kind of our early understanding of infectious disease and microorganisms to where we are now. So to start off, let's think about, early concepts of disease. Humans understood that certain foods became spoiled or inedible, that if you ate certain things, you'd get sick. But beyond that, most diseases were attributed to curses or demons, or even, sometimes it would be said that you or your family had done something wrong, and that's why you were sick. Even the disease malaria, for example, it has the name malaria, which comes from the Italian for bad air. It was thought, okay, well, you encountered some bad air and that's why you were sick. We know now that malaria is caused by a protozoan parasite that is carried by mosquitoes. So it was understood that there was some sort of transmissible material, like Black Plague and smallpox. There was some recognition that those things could be passed from one person to another, but there was also this underlying belief of spontaneous generation, that things could just appear out of nothingness, that there wasn't necessarily a cause or a pre-existing organism. So we start there, and we go into the early age of enlightenment, and we have Robert Hooke. Robert Hooke was a scientist, and he had some simple microscopes, and he was the first person to use the word cell. He published a paper entitled Micrographia, where he described the different things that he looked at with his microscope. And one of the things that he looked at was cork. So you can see this drawing on your screen. These are some of his original drawings of very thin slices of cork bark. Now, cork bark, you may know, is very, very light. And even though it appears solid, it is filled with all these spaces in it. And when you look at it under the microscope, you find that those spaces are actually where the cells that had made up the plant have died away, and they've left behind only the cell wall or the cellulose of the plant. So you see these very regular shapes in that cork bark. So he was the first person to really give us that term to describe kind of what we call the building blocks of living things. Another scientist by the name of Antony van Leeuwenhoek, he took Hooke's work and extended it even further. He refined the microscope that Hooke was using, made it even better, and he continued to look at microorganisms. And what's really cool about van Leeuwenhoek is he was not a scientist by trade, but it was a hobby for him. He did this for fun. And when he looked at a variety of different specimens under the microscope, he saw the first living microorganisms, those that were moving. He called them animalcules or animalcules. These are some of the drawings here, for example, and he looked at a large variety of things. What's really cool is that his drawings were incredibly accurate, despite the fact that his microscope was still pretty rudimentary. He looked at his own blood. He looked at pond water. He looked at fecal material. If you look over at these pictures, you can see he even looked at his own sperm. So he looked at a lot of different things under the microscope. You can see some very detailed drawings of rotifers and other unicellular microorganisms here. He just really spent a lot of time studying and looking at these things with his microscopes. So we started to understand that there were living things smaller than what we could see with our eyes. Remember, microbiology is the field in which we study living things too small to see with our eyes. So that's really where we started, right? Is by looking at different specimens where there didn't appear to be anything alive, but when we looked at them under the microscope, we could see them. Our next scientist that I want to mention is Edward Jenner. Edward Jenner was an English doctor, and he developed the first vaccine. The word vaccine comes from the Latin for cow, vacca, and he noticed that women who were milkmaids would get a rash illness called cowpox, but they would never get smallpox. Now, smallpox was a horrible disease. It killed lots and lots of people throughout history. And you can see in this picture on your screen, it gave individuals this really awful blistering rash, and it left lots of scars. He noticed that these milkmaids were not getting these scars. They were not getting smallpox. And it turns out that they got a similar illness that they caught from the cows themselves that he called cowpox. He used scabs from those milkmaids to make his first vaccine, and it turned out that if patients were inoculated or injected with the cowpox scabs, they wouldn't get smallpox. So that's really the first origins of our vaccines. So that's Edward Jenner. He's the father of vaccines. In the 1800s, we had Ignaz Semmelweis. Now, especially in this day and age, we take it for granted that we wash our hands. Ignaz Semmelweis was the first doctor to really demonstrate that washing your hands, particularly in the hospital, could prevent the transmission of disease. So he was a doctor, and he noticed that the patients in the maternity ward that were being served by the doctors were dying at a higher rate from what's called childbirth fever. It's really a strep infection of the uterus. He was noticing that the women who were being serviced by the doctors were dying at a higher rate than those that were being served by the midwives. So he investigated this, and he realized that the doctors, the thing that was really different between them and the midwives is that the doctors, because this is kind of the way that medicine was practiced back in that day, if somebody died from childbirth fever, they would take their body to the morgue, and then they would do a post-mortem examination, and they would do an autopsy. Well, the doctors, after they performed the autopsy, would not wash their hands. They'd return to the maternity ward and deliver more babies. The midwives didn't participate in that activity. So he proposed that maybe there was something that was being carried from those bodies to the living patients. So he suggested that they should wash their hands, that the doctors should wash their hands. And he did institute that in his particular ward, showed that it reduced infections down to the same level of the midwives. The sad part of this is 1840 where this happened, right? Hand washing did not become part of the regular routine in the hospital until the 1900s, right? So it took almost 100 years before this became common practice. While he was correct and he was kind of ahead of his time, right? It took a long time for scientists to accept that hand washing was in fact important. At this point, keep in mind, while we knew that microorganisms existed, we didn't understand that microorganisms caused disease. Right? So we're still kind of in that mindset that diseases were caused by curses and bad behavior. After Ignaz Semmelweis came Louis Pasteur. Now, Louis Pasteur did a lot of different things and made a variety of different discoveries. But one of the more important things that he did is that he showed that microorganisms did not spontaneously show up in liquid broths, but instead they came from the air. They came from particles that were brought into broths and into foods from the air. And what this did is it disproved what's called spontaneous generation, the idea that microbes could just appear. In the process of showing that microbes caused spoilage, he also developed a process we now call pasteurization, of gently heating milk and gently heating wine to kill off the microorganisms that might be present in those liquids, and that increased the shelf life and prevented those liquids and foods from spoiling, right? So this starts to lead to the groundwork of the germ theory of disease. He also contributed to the field by developing several of his own vaccines, one against a disease that was called chicken cholera, another one against rabies. So Louis Pasteur, you could write a book alone about his contributions to microbiology. So at this point, we're really starting to understand microorganisms can cause disease, or they're at least somehow related to disease and spoilage. And we're starting to understand that we can carry them from place to place. Joseph Lister, this is where we get the name Listerine from. He was the first surgeon to use disinfectants in surgery. So he noticed that if he treated patients with wounds, if he treated them with a solution called carbolic acid, that would prevent them from getting infections. So he was really the first person to institute this practice, and it really did help to increase the survival of his patients, right? Robert Koch came after him, and Robert Koch studied a variety of different diseases, but one important one that he studied was anthrax. So anthrax, it occurs in humans, but it also occurs in cattle, and he showed that cows that had anthrax had this long rod-shaped microorganism in their blood, but cows who did not have anthrax did not. He was the first person to link an infectious disease with a microorganism, and he did this through a series of experiments where he isolated the bacterium that he called Bacillus anthracis, and then he was able to grow it in the laboratory and inject it in a test animal and get that disease to occur in the test animal as well. This set up a series of postulates we now call Koch's postulates for proving the cause of a infectious disease. And we'll talk more about that as we go through this semester. But what I want you to remember about Robert Koch right now is that he was the first person to link a microorganism with a disease. This finalized the germ theory of disease, right? So Louis Pasteur started the development of the germ theory of disease, and it was Robert Koch who finalized it for us by showing that microorganisms could cause anthrax. After Robert Koch, we had Paul Ehrlich. Paul Ehrlich was recognizing that microorganisms could be the source of a variety of different diseases, and he came up with this idea of what we call a magic bullet, that maybe there could be some way to actually cure these diseases that were being caused by microorganisms by having a drug or a chemical that could kill the microbes but not kill the patients, not kill the humans, right? So this was his magic bullet idea. He spent his career looking for his magic bullet. He did discover one important antimicrobial drug. It was a cure for syphilis. Unfortunately, this drug also contains arsenic, so it was very toxic. So you could cure somebody from syphilis, but it wasn't something that you would use in regular everyday practice because it had a lot of different side effects to it. But this discovery by Paul Ehrlich and his idea of the magic bullet eventually led to the discovery of antibiotics. So Alexander Fleming was the scientist who discovered penicillin, the first antibiotic drug that was used to treat patients, right? So this was in 1928 that he made the discovery. It wasn't until about the 1940s that penicillin was produced for widespread use. All right, and then finally, towards the middle of the 20th century, we had the discovery of viruses, right? So there was an idea in the early part of the 20th century that there was something besides bacteria and our protozoa that were causing diseases, but they couldn't be visualized. In 1939, the tobacco mosaic virus, which is a plant pathogen, was the first virus that could be seen with an electron microscope, and we were able to demonstrate that it was the cause of this disease in plants. So these are some of the major events that led up to where we are today. Now, there were some other important things that happened in the 20th century. So we discovered a variety of different microbial causes of disease, lots of different species of microorganisms associated with very specific diseases, from malaria to syphilis to AIDS, for example. We were finally able to tie a specific microorganism to those diseases. We also discovered that DNA was the molecule of inheritance. Antibodies were discovered, and our understanding of the immune system became greater. As we got to the latter half of the 20th century, once we knew that DNA was the molecule of inheritance, we started manipulating the DNA and engineering the DNA. In the late 20th century, 1980s, 1990s, we discovered prions. Prions are a really unique type of pathogen. They are not viruses. They are not living things, but they're infectious proteins. So they're pretty interesting. And then another very important discovery of the 20th century is PCR, the polymerase chain reaction, that really revolutionized the way that we study DNA and we diagnose diseases. And we're going to talk a little bit more about PCR as we go throughout the semester. But all of these different discoveries led us to where we are today. We know that microorganisms cause diseases, and we're really focused now on understanding how they cause disease, what are their different mechanisms for causing disease, but also, remember I said in the last lecture, we're learning about those microorganisms that aren't pathogens, but that are living in our bodies, right? Our microbiome. We're starting to understand what is the role of those organisms in our health. So there's still so much to be discovered in the world of microbiology, and I hope you're as excited as I am about learning about these microorganisms and what they do.